Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear students, today we are starting with the one new chapter of cell biology, that is cell cycle and cell division of class 11th. In this chapter, we are going to discuss first cell cycle, and then we will be going to talk about the cell division. And in cell division, we will be discussing about the mitosis and meiosis. So cell cycle first we are going to understand. See, as we know that we humans have a lifespan. Same way, each cell has a certain life period and during which it carry out its growth activity and ultimately divide into two. So this time period from a one cell to become a two, and whatever the changes occur during this two within the cell, they are simplified or expressed in the form of cell cycle. So first question is, what are the characteristics of living cell? We know that every living cell has a capacity of growth and reproduction. First, every living cell will show a growth, means increase in protoplasm. And then for reproduction, they will divide into the two. So here you know that every individual has this properties of in every living cell as well. So all cells reproduce by dividing into two, with each parent cell giving rise to the two daughter cells. Every cell, if it is reproducing, it is going to produce a two daughter cell from the parent cell, and in this several process take place, and these newly formed daughter cells can themselves grow and divide again, and these newly formed cells are similar to their particular parent cell as well, and due to this division or reproduction property, they give rise to a new cell population. And this new cell population or mass of a cell or a cell population that is formed by the growth and division of a single parental cell. So if one cell is there, it divide into two, two cell will divide into four, four cell will divide into eight, and so on. The division will keep continue, and from a one parental cell, a mass of a cell or a new cell population will be formed. So what is the cell division? That first we need to understand. So cell division, if we talk, it is a process by which a parent cell divide into the two or more daughter cells. Like this, if it is a one cell, it divide into two, then this will divide into again two, and so on. That can keep on going. So that is division. Obviously, division is of two types that we will discuss. Now we are going to discuss first what is cell cycle. Okay. So if we define it, the cell division is a very important process in all the living organisms, dear students, and that is important for the reproduction or increase in the body size, increase the body number of cells. And during the division of cell, DNA replication or copying of DNA we can say occur, and cell growth also take place. DNA replication is a copying of a DNA, and it is a process of producing two identical replicas or copies from a one molecule of parental or original DNA molecule. So here this is the showing a process of replication, how the replication occur and how this process take place. That is not our concern in this chapter, that we will discuss in one separate chapter in class 12th, that is molecular basis of inheritance, where we will study how the process of replication take place. But you have to remember that in cell cycle DNA replication take place once. So this is what showing a DNA replication. Now what is cell cycle? If we talk about the cell cycle, it includes the sequence of events by which a cell duplicates the genome. It includes the sequence of events during which a cell duplicates a genome or a DNA or copy it and synthesize the other constituent of the cell, other organelles we can say, and eventually divide this cell into the two daughter cells. Then this whole sequence of events are termed under the cell cycle. So what it includes? The replication of DNA, synthesis of all the other constituent of the cell like cell organelle, cell components, and ultimately division into the two daughter cells. And these two daughter cells will be equal, are identical to the parent cell. Okay, so this is the cell cycle. So total duration of a cell cycle is a particular time period. So total duration varies from organism to organism and from cell type to cell type. If we talk about a yeast, cell, it takes 90 minutes to divide or complete the cell cycle, whereas E. coli takes 20 minutes to complete its cell cycle, and human cell takes 24 hours. See, so this much time is required. So time duration of the cell cycle varies from organism to organism and cell type to cell type. Now, this is the cell cycle which is shown in the diagram, and it includes several phases. So when we talk about the phases of cell cycle, dear students, we know that cell growth is a continuous process. Actually, there are no separate phases, but these phases are, this cell cycle, whole cell cycle is divided into different phases for our study purpose. So cell growth is a continuous process, and DNA synthesis occur only during one specific stage in the cell cycle. DNA synthesis occur only during one specific stage in the cell cycle, that is a S phase we will study. And the replicated chromosome, or we can say a DNA, are then distributed to daughter nuclei by a complex of series of events during the cell division. So this will occur during the cell cycle. So these events are themselves under genetic control. All the events which occur during the cell cycle, they are under genetic control, they are under the control of DNA. And a wide range of mechanisms that are used by a cell to increase or decrease the production of a specific genetic products, proteins, or DNA, that is under the control of DNA or genetic control. So it is important for the regulation of all the things, or regulation of the cell cycle. We will study that in the end of this chapter, or in the end of this lecture. So if we talk about the phases of cell cycle, you can see in this diagram the different phases of cell cycle are divided into the G1, S, G2, and this whole phase from here to here, it is comes under M phase, and sorry, up to this one, and this is the G0 phase. Here up to this is a M phase, and this is G0 phase. So such phases are there. So human cell in a culture divide approximately once in every 24 hours. We have studied that it takes 24 hours. And the duration of the cell cycle varies from organism to organism, also from the cell to cell. If we take example, yeast cell, we have studied that its body progresses its cell cycle in about 90 minutes, E. coli 20 minutes, and human cell 24 hours. So the cell cycle is divided into two basic phases. All the events of the cell cycle are divided into two basic phases. And when we talk about these two phases, they are interphase and we can say the M phase, that is mitotic phase. See, interphase and M phase. If we talk about interphase include a G1, S, and G2 phase, and M phase. Actually, M phase is the actual dividing phase. Here you can see this is G1. When cells start dividing or its cell cycle, first enter G1, then S, then G2, then M phase, and ultimately divided into the two. So this G1, S, and G2, this G1, S, and G2, they are the preparatory steps which are kept under the interphase. But M phase is the phase of actual division where mitosis or meiosis take place. So M phase is often called a phase of apparent division, means where the division is visible or actually it is occurring, and represent the phase where actual cell division or mitosis begin. It represents a particularly we can say a phase when the actual cell division or mitosis begin. So interphase represents the phase between two successive M phases. Interphase represents the phase between the two successive M phases. And this interphase, if we talk, in human cell, it occurs once in every 24 hours. Cell division occurs once in 24 hours, and cell division or M phase takes only 1 hour. So if cell division or M phase is taking only 1 hour, the 23 hours are of interphase. So 23-hour interphase will take 23 hours for the division in case of the cell cycle of human cell. So the interphase takes more than 95% of the total duration of the cell cycle. Yes. Now M phase starts with the nuclear division first. Then nuclear division will followed by the separation of the daughter chromosomes, that is karyokinesis, that is the nuclear division itself. And during nuclear division this occurs, so the cell nucleus get divided. Then the end with the cytokinesis, that is a cytoplasm division. Okay, so two things will happen during the M phase. One is karyokinesis or nuclear division, and then the cytoplasm division. First nuclear division will take place, and then cytokinesis will occur where the cytoplasm get divided. So first we are going to talk about interphase, then we will be doing to talk about the M phase. So the time during which the cell is preparing for the division. See, division actually occur in which phase? M phase. Yes or no? You can see here the division is going to occur mainly in this phase. So this G1, S and G2 are the interphase, means they are the preparatory phase. So it is a phase during a which or a time during which the cell is prepare itself for the division. By undergoing both a cell growth, it undergoes a cell growth and DNA replication in an orderly manner. In a proper sequential manner, this interface will be continued. Okay. So the interphase is a phase of non-apparent division of cell. Means during the G1, S and G2, the division or interphase is a phase of non-apparent division of the cell. Means during which actual division is not taking place, it is a preparatory phase. It is also called a resting phase. Okay, and the interphase is divided into three further phases. That is a G1 phase, which is known as gap 1 or growth 1 phase. Then S phase, which is a synthesis phase, and the G2 phase, which is a gap 2 phase. So what is G1 phase? First, we are going to talk about the G1 phase. So G1 phase correspond to the interval between the mitosis and the initiation of DNA replication. Why? Because DNA replication takes place in the S phase. Okay. This division takes place in the S phase, and this G1 phase comes before the S phase and after the M phase. So it is between the mitosis or M phase and the DNA replication. Why? Because DNA replication occurs in S phase. So during G1 phase, we will observe that what will happen. During this, the cell is metabolically active and metabolic activity will be observed. Cell will continuously grow and the replication does not occur. We remember replication of DNA does not take place. Now, what other activities takes place in G1 phase that we have to understand in a very well, that need to be understood very well. So during G1 phase, if we talk, it is occurring after the mitosis or the M phase, so we can call it as a post-mitotic gap phase. And during post-mitotic phase, gap phase, we have studied that cell grow to its maximum size. But DNA replication does not occur, so no change in DNA content. DNA content will not increase or decrease, it remains same as it is in the initiation of the phase. It will be similar at the end of the phase, that is the DNA content. Means if DNA content is 5 picogram in the initiation of the G1 phase, then it will be remain as a 5 picogram after the completion of the G1 phase. But the other things like new proteins and RNA synthesis will take place. The rRNA, tRNA, and mRNA. rRNA is a, we can say, ribosomal RNA. tRNA transfer RNA, and mRNA we can say messenger RNA. These all are RNA which are transcribed or through transcription they are produced from the DNA. Okay, so transcription will occur. Then the after the G1 phase in S phase, the replication is going to occur of DNA. And for replication of DNA, nucleotides are required. So nucleotides, amino acids for the protein, and energy ATP are formed or synthesized during the G1 phase. And when a cell, so much activities which are taking place during this G1 phase, it is going to take maximum time of all other stages. Maximum time. If it is approximately, we can say 24 hours in a human cell, it takes 9 to 10 hours during the G1 phase. Now it is also considered that G1 phase is considered as the most variable phase. It is the most variable in length, due to which the time of cell division differ in cell to cell. From one cell to another cell, the time duration is different because there is the most variable. If the cell particular cell cycle time period is less, or it is a frequently dividing cell, then the G1 time period of G1 phase is less. But if the it is a it is taking a long duration for the division, then the time period of G1 phase it will be more. So in a less frequently dividing cells, the time duration of the G1 phase is lengthy. Now, in G1 phase, at the end of G1 phase, when a cell reaches, if we talk about a G1 phase, when the cell reaches a stage where it will divide even under the stress condition. Means if at the end of G1 phase, if there is a no condition, it is not a proper condition, then it should enter the G0 phase. It should exit the G1 phase. Okay. But in spite of the state condition, it is going to divide. Then we call this end part of G1 phase as the end phase. And we remember one of the most important point of this G1 phase, that is the majority or maximum or most of the cell organelle formation or duplication take place during the G1 phase. It is important points you will find in many questions. So what is S phase? S phase comes after the G1 phase, and it is called as a synthetic phase, which marks the period during which DNA synthesis or replication take place. During which what will happen? DNA synthesis or replication will take place. So during S phase, the replication of DNA is going to occur. Then what other things take place that we understand? So the if DNA replication will occur, the DNA content will increase. So the amount of DNA per cell will double. If the initial amount of DNA is 2C, then it will increase to the 4C. Means if I say if it is the initial amount of the amount is 2 picogram, then it is going to be converted into the 4 picogram. Means the amount of DNA is going to be double. But there is no increase in the chromosome number in that space. DNA get replicated, so number of chromatid will increase, but the number of chromosome is not going to increase in the S phase. So diploid chromosome number remains the same even after the S phase. If the number of chromosomes are 46 in the initiation of the S phase, it will remain 46 after the completion of the S phase. But if the DNA content at the initiation of S phase is 5 picogram, then it is going to be 10 picogram after the S phase. In animal cell during S phase, we will see that DNA replication begins in the nucleus. The centriole also duplicate in the cytoplasm, because in plants centriole are not present. So in animal cell, the centriole duplication takes place in the cytoplasm. And apart from this, one protein is also synthesized during the S phase, which is known as histone protein. And this histone protein helps to coil the DNA, or we can say DNA get coiled around the histone protein and condense and form a chromosome. So for the formation of a chromosome or condensation of DNA or a chromatin material, the histone protein is required, which is synthesized in this S phase. Now S phase is also known as invisible phase. It is also known as invisible phase of cell cycle, because replicated chromosome are not visible in this state. Since replicated chromosome number is not going to replicated chromosome means what? See, initiation of S phase, at this phase they have a single chromatid, but when at the end of S phase the chromosome have a two chromatid, means here one DNA copy is there, that DNA copy get replicated, and one DNA copy remain here, one DNA copy remain here, and they get associated with the centromere region. So this way, the particular chromosome get replicated, or we can say them a replicated chromosome or chromosome with the two chromatid. Okay, but chromosome number is not going to increase in the S phase. After S phase, the G2 phase will come. And what is it? So at the end of the S phase and the starting phase of the pre-mitosis, the G2 phase will come in between these two. Okay. So it is the end of the S phase and the starting phase of pre-mitosis. In between this, the G2 phase will lie. So during G2 phase, if we talk, it is a gap 2 phase or growth 2 phase. Some of the important activities takes place which are necessary for the further M phase. So here some of the proteins are synthesized, and in this one important protein gets synthesized, which is known as tubulin protein. Dear students, be remember, why tubulin protein is responsible for the formation of the microtubule, and microtubule will form the spindle fiber and astral ray. And we will study that astral ray and microtubule are important for the division during the S phase, during the M phase. Sorry. Okay. So these proteins are synthesized. Then cell prepares for mitosis. Okay, cell is going to prepare for the mitosis, because after G2 phase, immediately the mitosis or meiosis is going to take place. So, and cell growth continues here also. So if we talk about the G2 phase, it is occurring before the M phase. So we can call it as a pre-mitotic gap phase. What we can call? Pre-mitotic gap phase. Now, So in this pre-mitotic gap phase, here what happens? The cytoplasmic organelles such as mitochondria, chloroplast, and Golgi complex are double. This mitochondria, chloroplast, and Golgi complex are double. Actually, most of organelles are already get synthesized in the G1 phase, but these organelles are also getting synthesized one more time in the G2 phase. Then apart from this, the synthesis of some RNA and proteins continues, and here the cell contains the double amount of DNA than the cell having DNA content at G1 phase. If at G1 phase the DNA content is 10 picogram, and then at the G2 phase it is going to become a 20 picogram. So now in G2 phase, the cell prepares itself to enter the M phase, because after G2 phase, immediately M phase is going to occur. Now in the adult animal cells, some cells do not divide. Okay. See, now if a cell want to divide, then it will continue what a G1 as G2M, G1 as G2M cell cycle. But if some cells do not want to divide, suppose in an adult animal cell, some cells do not want to divide, then it will going to enter the G0 phase. So we say that G is the cell at a G1 phase exit the G1 and enter the G0 phase. And such cells which enter the G0 phase are the examples are the heart cell and the nerve cell. So this G0 phase is also known as quiescent phase in the cell cycle. So the cells do not want to divide further after G1 phase, then enter to an inactive stage called a quiescent stage of the cell cycle. And the quiescent stage of the cell cycle is the G0 phase of the cell cycle. So cell in this stage remain metabolically active, and cells not proliferate unless it is called on to do so depending on the requirement of the organism. If there is a requirement of division, then the cell will exit this G0 phase and come again and the G1 and continue the cell cycle. But if there is no need of extra cells or much cells, then the cell will exit the G1 and enter the G0 phase. So many other cells divide only occasionally as needed to replace the cells that are injured or dead, means they are in a state of what we can say G0 temporarily. So in animals, mitotic cell division is only seen in diploid somatic cells, okay, and in plants, the mitosis cells division is seen in both haploid and diploid cells, okay. So now we are going to understand the mitosis. Before that, we are going to understand the regulation of the cell cycle, okay. So whatever we have studied about now, that is about the interface, G0 phase, G1, S and G2, whatever the activities takes place during this. And after G2 phase, M phase will takes place. But before studying the M phase and mitosis and meiosis, first we need to understand the regulation of cell cycle. And if we talk about the regulation of cell cycle, it is known that decision of a cell to divide occur in G1 phase. Occur in G1 phase. And here it is known that if a cell is not to divide, if a cell do not want to divide, it enter G0 phase by exiting or exit of G1 phase. But when a condition change, the cell can enter the back to the G1 phase. Now here this regulation and further regulation is regulated by the checkpoints. So checkpoint or restriction points of cell cycle. And if we talk a checkpoint or restriction point of a cell cycle, the first restriction point is located at G1 to S transition, and second restriction point is located in at G2 to M transition. These two are the checkpoint. So here, suppose this is the cell cycle. And if this is the cell cycle, here the G1 phase is there, here G0 phase is there, here M phase is there, here S phase is there, here G2 phase, and here M phase. So one restriction point is located in the G1 to S transition, and another restriction point is G2 to M transition. So first checkpoint, and this is second checkpoint. So these checkpoints are located, and it is known that the first checkpoint, that G1 to S, is the major checkpoint, and it is known that once the cell crosses, and it is known that once the cell crosses restriction point, once the cell crosses the restriction point, cell cycle get completed. Okay, so these two restriction point are there. Now how this restriction point occur? So this restriction points, this passing or crossing of restriction point is regulated by cyclin dependent protein kinases. Cyclin dependent protein kinases. Okay, so we can write CDK, cyclin dependent kinases. Okay, now it is known that cyclin are protein that activate cyclin dependent protein kinases and regulate cell cycle. So these are cyclin are important for the activation of this CDK. Now it is known that this cyclin are of two types. One is G1 cyclin and one is M cyclin. So this cyclin are present in the, particularly we can say a cytoplasm. Okay, and it is known that G1 to S transition is carried out by association of G1 cyclin, G1 cyclin with cyclin dependent kinase, that is CDC2 kinase. And when this bind with this cyclin, the activation occur and the cell will crosses this checkpoint from G1 to S, because when they associate, they initiate the replication of DNA, and replication started. If G1 cyclin associated with the CDC2, the replication will initiate. And if replication has started, means S phase is started and it completed the G1 phase. So same way, if you talk another thing about the another checkpoint, it is known that G2 to M transition, G2 to M transition is triggered by MPF, that is maturation promoting factor formed by fusion of M cyclin and cyclin dependent kinase type, that is CDC2 kinase. Okay, so this binding occur and then it will cross the G2 phase to M phase. Okay, so these are the things where you will see that the activation is occurring. Okay, so this is about the what, a particularly regulation. Now we are going to talk about the factors which determine whether a cell has to divide or not. Cell has to divide or not. And this cell has to divide or not, the factors are: number one, surface area to volume ratio. So if surface area to volume ratio, if you want to understand that how it is helping the cell to divide or not, suppose we are taking one cube and its length, breadth and width is, we can say, particularly 3 centimeter. Okay, and if I take a same cube with the length, breadth, so if here the length, breadth and width is 2 centimeter and the another cube is there whose length, breadth and width is 1 centimeter, So if we talk about surface area to volume ratio, the cube surface area will be calculated by 6a square and its volume will be by the a cube. So if we take a3, then 3 square is 3. If we talk 6 and a is equal to 3 and here 3 cube. So 3 square is 9, 9, 6 9 ja 54 upon 3 cube. 3 3 ja 9 3 ja 27. So 27 divided by 2. So surface area to volume ratio is 2 for this 3 centimeter height. But for this, if we talk 6a square upon a cube, then it is a 2 square. So 6 into 4 upon 2 cube 8. 6 4 is 24 upon by 8. So it is going to be 3. And if it is a 6a square, so 1 square upon 1 cube, so it is a, we can say a square 6, 6 upon 1 cube is 1, so it is going to be 6. So surface area to volume ratio, as the size is decreasing, the surface area to volume ratio is increasing. So surface area to volume ratio will increase as the size decreasing, and it is considered that the surface area to volume ratio increase, it increase, then cell divide. And if surface area to volume ratio, sorry, it decrease, okay, then cell divide. Because if it decreases, then the size is increasing. And if size increases, then it is going to, growth is taking place. So after the growth, the cell will divide. And the another factor is karyoplasmic index. So this karyoplasmic index or nucleocytoplasmic ratio, we can say, this is the formula for this is Vn upon Vc minus Vn. And here the Vn is equal to volume of nucleus. Vc is volume of cytoplasm. Okay, so volume of nucleus upon volume of cytoplasm will determine this thing. So during division, growth of cytoplasm occur. And if the growth of cytoplasm occur, the cytoplasm is growing and nucleus is smaller, so means during division, karyoplasmic index Ki decreases. Okay, so this is the what a karyoplasmic index, and this is the two factors which are responsible for the determination that division will take place or not. Okay, so I hope you understood this. This is all about the cell cycle. Now about the M phase or mitosis and meiosis, we are going to discuss in the upcoming lectures. Okay, I hope you understood. Thank you.